we have, sorry, what did I do? I don't know how to get rid of those subtitles. Does anyone know how to do it? Give me a hint. It's kind of irritating to look at. I don't know what I clicked. Oh, yeah. Okay. So cytokines like tumor necrosis factor, remember this is one of the potent pro-inflammatory cytokines. So when it's produced by your white blood cells, your activated white blood cells, for example, your macrophages or dendritic cells, it can travel to through the circulation to the hypothalamus, where it increases the temperature set point. And this is a protective effect in your inflammatory response, because this higher temperature is thought to inhibit growth of pathogens and to increase effectiveness of the immune response. So this patient will present with a fever, okay? For your paracrine action, an example of this, and we'll get to see details of this when we talk about our cell-mediated hypersensitivity or cellular hypersensitivity or type 4 hypersensitivity, where you have your macrophage in this case presenting antigen to your T cell, and then the T cell produces interferon gamma, which acts on the macrophage that was presenting antigen to it to activate the macrophage, okay? An example of autocrine action is where we have an activated T cell. Once it has recognized antigen that is presented to it, producing interleukin-2, and then the interleukin-2 comes and binds to the interleukin-2 receptor on the same T cell, which now will cause it to proliferate, okay? So you get more production of that particular clone of T cells. The other property of cytokines is redundancy, which basically means you can have more than one type of cytokine inducing or having the same effect, okay? And this is shown in a picture here, where you can have your CD4 positive T cell or your T helper cell producing different types of cytokines that would all activate your B cell. So you can have, it can produce interleukin-2, which can activate your B cell, interleukin-4 activates your B cells, interleukin-5 activates your B cells, and all of them will cause your B cells to proliferate. So this means if you, in terms of redundancy, if you are trying to block the activation of B cells, inhibiting just one of these cytokines wouldn't produce your desired effect, because you can still get your B cell activation through any of the other cytokines. They also act in synergy, where you get potentiation of the effect by the combined action of two different cytokines. So an example here is where you have interleukin-4 and 5 acting in synergy or acting in combination to activate your B cell and induce class switching of your B cell from IgM to IgE, although interleukin-4 is the most potent of the two in causing class switching, but the combined effects of both can occur. So the clinical aspects of this, as I've already mentioned, is the fact that if you attempt to block one cytokine, it may not produce your desired effect, because you may have that effect being caused by another cytokine. And an example here is in rheumatoid arthritis, where they have tried to use, or actually they do use antibodies that block tumor necrosis factor. Remember, I've alluded to this as a pro-inflammatory cytokine. So they've used monoclonal antibodies to TNF or anti-TNF to stop that inflammation in rheumatoid arthritis that causes joint damage. But this will only block TNF. You have other pro-inflammatory cytokines like interleukin-1. So using the anti-TNF on its own is not enough to completely get rid of the inflammatory processes. The other property of cytokines is pleiotropism, which means you have one cytokine acting on different cell types, or many cytokines acting on different cell types. An example given here is interleukin-4 produced by your T helper cell, We've already mentioned it can activate your B cells, cause it to class switch or differentiate into plasma cells, and then class switch the immunoglobulin isotype. The same interleukin-4 can also cause or have effect on cytotoxic T cells and also on mast cells, okay? So in terms of the clinical context, this means that if you block the interleukin, or rather that particular cytokine that has an effect on many cell types, you may get effects that you do not really want. And an example given here is the use of interferon alpha in the treatment of chronic hepatitis B virus infection. So interferon alpha has some pro-inflammatory effects. So not only will it we will produce the desired effect of blocking viral replication. Remember, your type 1 interferons alpha and beta usually inhibit viral replication, so they're important in the immune defense to viral infections, but it can also induce an acute phase response, okay? And this is that inflammatory response that usually makes patients feel unwell, so your fever and so forth. So patients would feel unwell because of the treatments that you're giving them, and that is just because of that pleiotropic effect. The other property of cytokines is that they can be antagonistic, so they can inhibit each other. So an example here is interleukin-4 activating your B cell and causing it to class switch, but interferon gamma acting on the same B cell will produce the opposite effect. It will block the class switching, okay? Another example would be where you have your anti-inflammatory cytokines inhibiting the pro-inflammatory cytokines. So the two major anti-inflammatory cytokines are interleukin-10 and transforming growth factor beta. So they'll produce the opposite effect of your pro-inflammatory cytokines, your interleukin-1, 2, and tumor necrosis factor. Okay, the other property is that they act in a cascade, okay? And so this is the example I alluded to, where you have your macrophage presenting antigen to your T helper cell, and then it produces interferon gamma that activates your macrophage. But in this case, you can also have your macrophage producing other cytokines like interleukin-12 that activate your T helper cells, which now produce more cytokines. Okay, so you're getting that sequential activation of cells in a cascade. Okay, in terms of nomenclature, which probably ties in with the classification, there are very many cytokines. I guess new ones get discovered all the time. So far, more than 300 cytokines have been described. It's difficult to know all of them. We usually just allude to the commoner ones in terms of the type of immune response we're talking about. And because there are very many, we don't have a standard naming system, which would include all of them, that would be all inclusive. So I'll just highlight the commoner and largest groups. So one is the interleukins. This is the largest group. They're the ones we abbreviate as IL and then a hyphen and a number. So they were named so because they were thought to act between leukocytes, so between one white blood cell and another, but it's been shown that they have effects on many cell types. And they are numbered according to the order of discovery. So from interleukin 1, 2, 4, 5, 7, and so forth, okay? And they all have different functions. For example, interleukin 7 is important in the process of hematopoiesis in the bone marrow, okay? The other group are the interferons, also named after the presumed function. It's thought that they interfere with viral replication. So interferons are very important in the early immune response, the innate immune response to viral infections, and specifically the type 1 interferons. The type 1 interferons are interferon alpha and beta. The other interferon is a type 2, which is interferon gamma. That one is more important in cell-mediated immune responses, okay? They're potent activators of the immune system. Actually, the interferons are very important. It's been shown that there's a group that does a lot of research on primary immunodeficiencies, And also have like phenocopies. So primary immunodeficiencies usually would mean there's a certain gene defect in any component of the immune system, including your cytokines. Phenocopies would be where there's a gene defect, but it's a somatic probably defect. So they've seen that people who have antibodies against the type 1 interferons tend to develop very severe COVID-19. So it's not really a PID, it would be more like a phenocopy because there's no problem with the gene, but it presents as if there's a problem with the interferon alpha. So these patients, because they have antibodies that block your interferon alpha or beta, they're not able to limit the early viral replication when they have infection with SARS-CoV-2, so they get overwhelming disease. The other group are the tumor necrosis factor. This one named so because in vitro, they were shown to induce necrosis in cancers, not really in vitro, but in animal studies, when injected into animals at high concentration. However, this has not been shown to be a very potent effect in humans. Otherwise we'd be able to get rid of cancers very easily just with production or with injection of tumor necrosis factor. So that is why they were given that name, but that effect is not as potent. So they're very important in inflammation. That TNF is pro-inflammatory. And then we have colony stimulating factors. I won't go into details of this. I'm sure you know all about this. These are the ones that are important in hematopoiesis, like your GCSF, your GM-CSF, and so forth. So this one's just as the name suggests, they help in proliferation of certain cell types. Chemokines, as I mentioned, are the chemoattractant cytokines. So they stimulate cell migration. And the naming is based on the number and spacing of a specific cysteine residue. And you've seen all these types of names like CCL2, CXCL8. They're also clinically important, as you've seen in certain diseases, like in HIV, people who have mutation in some of the chemokines or the receptors become resistant to viral entry. In terms of classification, the various, as I mentioned, there's not one single classification system that would be all-inclusive of all the more than 300 cytokines described, but generally they can be classified according to structure or according to the function. So according to structure, it's basically that biochemical structure. So you have the hemopoietin family. So the hemopoietin family would include both the colony stimulating factors and other cytokines that are important in proliferation of these cells, okay? So like the interleukin-2, remember, it would cause proliferation of T cells, for example. So it's important, interleukin-4, interleukin-7, okay? And then there's the interferon family. So this structural classification just means they have some similarity in that biochemical structure. So the interferons are interferon alpha, beta, and gamma. There's the chemokine family and the tumor necrosis factor family. When you classify them based on function, it's just as the name suggests, what do they do? So there are some that are growth factors. So for example, the hemopoietins would be growth factors. The colony stimulating factors would be growth factors because they cause not really differentiation, but proliferation of cells. And then the other class based on function are those that are pro-inflammatory, okay, like the TNF, the interleukin-2 and so forth. And then there are the anti-inflammatory agents. The anti-inflammatory ones are interleukin-12 and 10 and transforming growth factor beta. And then there are those that are polarizing agents, which means they drive cell differentiation into a certain cell type, or they cause cell specialization. And I'll just talk about a few of these. So even the ones that would cause, for example, a B cell to differentiate into a plasma cell, they're polarizing that cell to a particular phenotype, okay? So this is just a table summarizing some of the cytokines and the cells that secrete them. You can see most of them are white blood cells, but you have a few other cells that are not white blood cells. that could secrete them, but majorly they're secreted by white blood cells, but you can have like endothelial cells and epithelial cells, and then their target effects. I like this classification that classifies or differentiates cytokines of the innate immunity, meaning they're majorly produced by the innate immune cells. And you can see these are basically your antigen presenting cells, monocytes, macrophages, and dendritic cells. And then we have the cytokines of adaptive immunity. So these are the ones that will be produced by your T cells, okay? What I just want to highlight is that the ones of the innate immunity tend to be the very potent pro-inflammatory cytokines, okay? Like your interleukin-1, TNF-alpha, interleukin-6, okay? And then, of course, the interferons, okay, which are important, alpha and beta, which are important in inhibiting early viral replication. Remember, it will take, when you have a pathogen gaining entry into a host, it takes some time to activate the adaptive immune response. So at that immediate time of exposure, you need your innate immune response to try and prevent proliferation of this pathogen. We can see the different effects of these cytokines. And I just want to highlight what the cytokines we've heard about in the cytokine storm in SARS-CoV-2 tend to be the innate immune, the cytokines of innate immunity. I'm sure we all know, we've heard a lot about interleukin-6 and the use of tocilizumab to try and prevent severe disease or treat, let me say treat those who present with severe SARS-CoV-2. It's a monoclonal antibody that blocks the receptor to interleukin-6, okay? So you can go read further on these cytokines and their target effects. The ones of adaptive immunity, as I've alluded to, they're produced by T cells, and they'll act on either the T cells or even some of the cells, innate immune cells, okay? So I think these are, I would expect you to actually know this small list of cytokines. These are the ones we allude to very commonly, okay? We'll come to see that interleukin-4 and 5 are very important in allergic immune responses, so I expect you to know what they do, okay? Transforming growth factor beta is important in immune regulation. It's an anti-inflammatory cytokine. Interferon gamma is the one that activates your macrophages. It's important in cellular immune responses, especially against intracellular pathogens, okay? So at a minimum, you must know these cytokines. This is another classification just based on function, as we talked about. You have the pro-inflammatory cytokines like TNF, interleukin-1, interleukin-6, and some of the chemokines. The anti-inflammatory ones, important are interleukin-10 and TGF-beta. There are those that inhibit viral replication, so interferon alpha and beta. The ones that activate macrophages or important in cell-mediated immune responses, interferon gamma. And then those that activate B cells, those that activate T cells, and then those that act on eosinophils and mast cells. These are important in allergic immune responses. Okay? So this is just a small list out of the 300. Okay, in terms of secretion, we've already said they're secreted by various cell types, including immune cells and non-immune cells. And we've talked about how cytokines have a very potent effects, and so secretion is tightly regulated. So most are only secreted when these cells are activated, and usually it's as part of the response to infection, okay? So they're not produced constitutively, okay? There has to be something that induces their secretion. Few are secreted constitutively, constantly at very low levels. And these ones usually are not the ones that have the potent effects, otherwise they'd end up causing adverse effects. In terms of the levels of secretion, it's variable. I guess this would depend on the amount of antigen that is triggering activation of these cells that secrete them. So you don't expect the same response all the time in every host that's producing these cytokines. So those of the innate immune system tend to be secreted at low levels, and they act over a short range, okay? So when there's proper regulation of the immune response, you only want them to be produced and act where your triggering antigen is. okay? However, if there's overwhelming activation, they can be secreted at high levels, and then they go and act in an endocrine fashion. And this is what we see in cytokine storm, okay? And because of this, they'll go and act on very many cell types. And this is why these patients with cytokine storm tend to have multi-organ dysfunction. You know, they have the infection in the lungs, but they end up getting kidney disease, they get brain injury, they get liver pathology just because of very high levels of cytokines being produced and the endocrine fashion in which they're acting. For the adaptive immune system cytokines, they're also secreted at low levels, and majorly they'll have paracrine and autocrine effects. Okay, it's rare for those of the adaptive immune system to be secreted at high levels. So even just in the cytokine storm, the implicated cytokines are those of the innate immune response. So interleukin-1, interleukin-6, and tumor necrosis factor. So because of this, when it comes to evaluation, it's very difficult to detect, to be able to measure the adaptive immune cytokines in a blood specimen, even if a patient is unwell. For those of the innate immune system, if a patient is having, you know, a massive secretion of the cytokines, we can actually measure them in blood. And one of the tests that is being done in some centers for patients who develop symptomatic SARS-CoV-2 is detection of interleukin-6 in patient specimens, okay? Because they are produced at a level that you'd expect it to be detected, and it's being secreted in blood for it to act in that endocrine fashion. That means it would be detectable in the lab. So secretion of cytokines is usually transient. We've talked about the proper regulation or tight regulation. So usually in response to infection, once the infection has been cleared, then the secretion stops, okay? And the other thing that occurs is you also get, you know, as the infection is resolving, you also get secretion of the anti-inflammatory or the inhibitory cytokines like interleukin-10 and TGF-beta. So this just brings back a balance, and you get to see a lot of the immune responses, just like any other response in the body. Even if you talk about hormone production, it's very tightly regulated so that we always try and maintain homeostasis, okay? In terms of the action, which also helps to balance the secretion, is that the receptors that the cytokines use to induce their action are also expressed transiently, okay? When you have, for example, if you have activation of a T cell and you get production of interleukin-2, you also get gene activation of the gene that codes for the interleukin-2 receptor. So when you stop, like the activation of the cell is reduced, then also production of the receptor is reduced, or expression of that receptor. So these are all checks and balances that help to prevent inappropriate activation of the immune system. I think this is another table that you can look at later, just listing some cytokines and their general effects. I won't go into details of that. I've talked about the fact that cytokines act via receptors. Okay? So if the receptor is missing, the cytokine cannot exert its function. And we'll mention some examples of primary immunodeficiencies where there could be a defect either in the cytokine or in receptor. So either way, you won't get action of that cytokine, and this can lead to lack of an immune response. Okay, we'll talk about that later. The receptors are classified based on their structural homology. If you remember this from biochemistry, about 6 different cytokine receptor families. You have the immunoglobulin family receptor, the hematopoietin receptors, which have 3 chains, an alpha, beta, and a gamma chain. And this is where your cytokine would bind. You have your interferon type of receptors, your TNF receptors, interleukin-17 receptors, and chemokine receptors, which act via G proteins. I won't really go into details of that, but I'm sure you know once you have binding, then these cytoplasmic tails are the ones that are important in transducing the signal to the nucleus, and then you get gene activation. Okay, so this is a bit more on the signaling molecules. So what happens after, I've already mentioned that most of the receptors are not expressed in high numbers on resting cells, but when the cells are activated, you also get upregulation of these cytokine receptors. Okay, so you get more of the receptor expression, and now they are spread across the surface of the cell. Once a cytokine binds, and this is the same thing we see with, for example, an antigen binding onto a B cell receptor. Okay, you get like clustering of different molecules on the surface of the cell, which helps to activate your intracellular signaling processes. Okay. So each family of receptors is linked to different signal transduction molecules. These are like your JAK and STAT proteins. I really won't expect you to know much detail of this, but this is just, I'm highlighting it so that you understand how, when the cytokine binds, how do we get activation of a certain gene, okay? So you have your cytokine binding to a receptor here. Usually the intracellular or the intracytoplasmic processes are associated with different tyrosine kinases, which become phosphorylated, and then they also phosphorylate like your, should I call them like adapter proteins or other proteins that you find intracellularly, which are able to, through these phosphorylation processes, these are able to transmit this signal from the surface of the cell into the nucleus so that you get a particular gene being activated and being transcribed, and then you get a particular protein being produced. Okay, I hope that is clear. I see something on the chat. Let me just see if it's a question before I proceed. Does obesity induce chronic low-grade inflammation have similar cytokine storms as an acute COVID infection? Probably not, because it's low-grade inflammation. In a cytokine storm, you're having uncontrolled inflammation that would make a patient acutely ill. So just the way as you have mentioned, if it's low-grade inflammation, a patient won't be acutely ill. But because there is inflammation, then there's probably some level of cytokine production going on, because that is what will induce and cause the continuous inflammatory processes. Okay, so what is the role of cytokines in the immune response? So one is initiation of inflammation. I'm sure you've gone through the inflammatory process. We introduced it in year 2. You've covered it in your general pathology. And you've seen for these cells to exit the circulation and come to the tissue where the triggering antigen is, they have to be guided. And usually it's the chemokines that are important in causing the attraction of these cells so that they can transmigrate from the circulation to the site, okay? They're also important in communication between the innate and adaptive immune systems. We've seen through the process of antigen presentation, also to activate these cells, you need cytokines to be produced. I've shown you, like where I was showing you the cascade induction. You get cytokines being produced by your innate cells that act on your adaptive or your T cell. Then the T cell produces cytokines that will probably activate your innate cells. They also cause T cell priming, okay? When your T cell recognizes antigen for it to be activated, that's the process of priming. They're important in development of T cell specialization. So this is also part of the polarizing effect of cytokines. You know that we have different subsets of T cells, okay? For example, if we talk about the T helper cells, that's the CD4 positive T cell. We have 3 types, the T helper 1, T helper 2, and T helper 17, okay? So there are different cytokines that would cause, for example, T helper cells to differentiate into either the 1, the 2, or the T helper 17 subtype. And then they're also important in winding down of the immune response, okay? This is why you have your inhibitory or your anti-inflammatory cytokines, okay? So just reminding ourselves the roles of cytokines in the innate immune response. These are the ones that will be produced by your macrophages, and they're usually the pro-inflammatory cytokines, as well as chemokines that would now, so it's the resident tissue macrophages that would be the first to interact with an inducing antigen, for example, a pathogen, and they start to secrete cytokines, which would cause the transmigration of neutrophils. So they recruit neutrophils and other immune cells to a site of infection. So they're responsible for causing acute inflammation. We've talked about the role of the type 1 interferons early in a viral infection. Okay, so they inhibit viral replication. They cause the already infected cells to become more susceptible to killing, and they cause the neighboring cells to be resistant to viral entry. Okay, and there are different ways by which they do that. I think in second year you went through the immune response to viral infection, so you can go and revise that. Very relevant right now because we're in a pandemic that is caused by a viral pathogen. So I mean, that is a highly examinable topic right now. So that's leakage I've given you. The other role I've talked about is in T cell priming, okay, as your T cells are being activated. So remember, to activate your T cells cannot recognize antigen in its native form. It has to be presented to it by an antigen presenting cell. But that as the first signal on its own is not enough to activate your T cell. You need a second signal, okay, which is what we call the co-stimulatory signal. So, for example, your CD40 acting with a being activated by the CD40 ligand, or CD80 or 86, the B7 molecules. Okay, so these co-stimulatory signals could include those molecules or even some cytokines. Okay, like your type 1 interferons or interleukin-1. So this basically just means you need a second signal. The CD40, CD80, or CD40 on your B cell and ligand on the T cell are basically, the CD40 would be a cell surface expressed cytokine, while the ligand would be one that is secreted, okay, because it's the one that comes in and acts on the CD40, which is the receptor, okay. I've talked about interleukin-2 and its interleukin-2 receptor being produced once you have activation of a T cell, so that you get proliferation of that T cell. That is another way of T cell priming, okay? So this is basically that second signal that occurs, and then the downstream effects after that. And we'll go into further detail of this. That is the clinical implication of this is very important when it comes to transplant immunology, because we'll talk about the different targets that can be blocked to prevent rejection responses. So in terms of polarization of your T cell or development of specialized T cells, I've talked about T helper 1 responses or T helper 1 T cells. So this occurs when you have an intracellular pathogen, which stimulates your antigen presenting cell. As it's presenting antigen to your T cell, it also secretes very specific cytokines. Usually it's interleukin-12 and type 1 interferon. So these cytokines will induce your T cell to secrete interferon gamma. And once they do that, they acquire a T helper 1 phenotype. So we call it like the pro-inflammatory phenotype, because it actually just causes activation of your immune cells. It's basically your cellular immune response, okay, which involves your CD4 cells and CD8 cells. So it can also activate your cytotoxic T cells. We've talked about interferon gamma activating your macrophages, okay, to kill intracellular pathogens. And you'll see in anatomic pathology that these activated macrophages are the same ones we call the epithelioid cells that are important in the immune response to certain infections, like that by Mycobacterium tuberculosis. Okay, they may also cause production of IgG by B cells, but the more important aspect is that you get more of a cellular immune response. Okay, and we'll see how, when we can't get rid of these pathogens that induce this kind of response, this leads to granulomatous inflammation. The T helper 2 responses favor antibody production, especially IgE. So these are responses that we see, especially with infection by helminths and parasites. And basically the cytokines that are produced will cause activation of a different transcription factor than the one we get in the T helper 1 responses. I really don't need you to know the details, that it's GATA3 that is induced. So what happens is that majorly you get production of interleukin-4, okay? We've talked about interleukin-12 and interferon gamma in the T helper 1 responses. In T helper 2, you majorly get interleukin-4 production, and this one is important in causing isotype switching of your B cells to IgE. And I think we've already alluded to the fact that immune responses against helminths are majorly caused by IgE production and activation of your eosinophils, okay? So these cells also secrete interleukin-5 and eotaxin. Interleukin-5 causes activation of and proliferation of eosinophils, while eotaxin is chemoattractant for eosinophils. So in terms of pathology, these T helper 2 type of responses are the ones seen in immune-mediated diseases where you have a majorly an antibody response, for example, allergies, which are mediated by IgE production, but there are also some autoimmune diseases which develop majorly because of T helper 2 responses, where you have, usually there should be a balance of T helper 1 versus T helper 2, but in this antibody-mediated immune diseases, you tend to get more of the T helper 2 responses predominating. T helper 17 responses, this is where you have generation of T helper 17 cells because of production of interleukin 23 induced by these pathogens, majorly extracellular pathogens like bacteria and fungi. So they produce interleukin 23, which activates your T helper cells, and then the T helper cells produce interleukin 17. Okay, so interleukin 17 attracts, it's very chemoattractant for neutrophils. So it attracts neutrophils, the site of infection. So you get a neutrophil inflammation. Interleukin 17 is one of the. Switch off your mic. Interleukin 17 is one of the recently discovered interleukins or cytokines, and it's been shown to be the one that is produced by despolarized T helper cells, so they were called T helper 17. The clinical implication of this is that there's a subset of primary immunodeficiency diseases where patients lack this production of these cytokines, or they don't have T helper 17 cells, and these patients tend to be predisposed to infection by extracellular bacteria and fungi. Okay. The other specialized T cell responses is in gut immunity, where remember you have the mucosa-associated lymphoid tissues. So here the T cells in the gut secrete TGF, or transforming growth factor beta. This is part of, remember we said this is an inhibitory cytokine. So it's part of the immune regulatory response, or immune tolerance, specifically oral tolerance. We expect that because what the antigens we take in food, yes, they're foreign proteins, but usually we don't mount an immune response against them. And this could be because of the action of TGF beta. And what this does is induces a class switch in the B cells that are found within the gut, or the mouth or gullet, from IgM to IgA, and then you get the IgA being secreted into the mucosa. And we know that that's important for mucosal immunity because it's secreted into mucosal surfaces. Okay. TGF beta can also induce T cells to become the T regulatory cells. And T regulatory cells, as you remember, they're important in balancing the immune response or causing immune suppression. They're also important in providing tolerance where you don't get a response against self-antigen. Okay, the other role we mentioned is in ending of the immune response. I've already talked about the fact that, once the pathogens levels become low, then you should get a reduction in the levels of the cytokines that are being produced, okay? Of course, you have less antigen available for presentation, you have less antigen available for recognition. So of course, you get less stimulation of your T cell, and because of that, now you get lower levels of cytokine production, less receptor expression. So that is the natural response. Once you get rid of the inducing agent, then you should start getting a waning in the immune response. The inhibitory or anti or immunosuppressive cytokines are important in ending the immune response. The ones I need you to remember are interleukin-10 and TGF-beta. So they just do the opposite of what the pro-inflammatory cytokines would do, okay? Okay, so now I'll just touch on some cytokine-related diseases, and you can go and read further on this because now you're doing your systemic pathology. So in certain bacterial diseases, there are some toxins which can activate a large number of T cells. And when these T cells are activated, they produce large quantities of cytokines. For example, your staphylococcal enterotoxin, or your toxic shock syndrome toxin that's also produced by your Staph aureus. These are super antigens, they can activate a large number of T cells directly, okay, without the need for antigen presentation. And this causes overwhelming production of cytokines, which for example, if they secrete interferon gamma, this activates macrophages, and the macrophages secrete a lot of pro-inflammatory cytokines. So you can end up with a cytokine storm. So the cytokine storm is not very specific just to SARS-CoV-2. It can occur in other diseases. For example, staphylococcal infection can also occur in infection by the influenza virus, okay? So it just alludes to the massive secretion of your pro-inflammatory cytokines, interleukin-1, 6, and TNF. And then because these are secreted in an endocrine fashion, they go and act on many target organs, and a patient will present with sepsis or septic shock. So with DIC and multi-organ failure. And we've seen all this in different types of cytokine storm, whether it's caused by SARS-CoV-2 or caused by another pathogen. The pathophysiology is the same, okay? So there's a similar form of pathology called the cytokine release syndrome that has been seen in some forms of immunotherapy. Probably we'll talk about immunotherapy later, where they use certain cytokines as therapy, for example, in cancer immunotherapy, okay, which can cause a similar sort of response, but it's usually mild. Our patients don't really get septic with this, okay? So I've talked about certain diseases where either you have a mutation in a certain cytokine, so of course you don't produce that cytokine, or a mutation in the gene that codes for that cytokine receptor. So even if you produce a cytokine, if its receptor is missing, the cytokine will not be functional. So one I'm mentioning here is mutation of the interleukin-2 gamma receptor chain. Remember, I showed you those the interleukin family of receptors where it has an alpha, beta, and a gamma chain. So there's a primary immunodeficiency where the gamma chain for the receptor is missing. And it's a common gamma chain, common to interleukin-4 receptor, 7, 9, 15. So, and remember I talked about interleukin-7 being very important in hemopoiesis, as well as interleukin-2. So what happens is that you actually don't get production of, or good production of your hemopoietic cells, so natural killer cells, T cells, and B cells, because they don't develop further because they're missing this cytokine receptor, okay? And this would cause a severe combined immunodeficiency. You can see the cell types that are missing. It's natural killer T and B cells. We'll discuss this further in the topic of primary immunodeficiencies. The other one is the CD40 ligand, okay? Remember, to activate your B cells can either be activated directly by an antigen, and then they would just, differentiate into plasma cells, but in this case, they'd only produce IgM, the default immunoglobulin. For you to get isotype switching, you need that germinal center reaction where you need a T helper cell to be activated by the same antigen that activated your B cell. Most of the time, this occurs in the context of the B cell presenting that antigen also to the T helper cell. Okay, I hope you remember that from your second year. So because you're activating your B cell, you need a co-stimulatory signal, and this is where your CD40 ligand interacting with CD40 occurs. So it's only in this context of your T cell activating your B cell that you would get antibody class switching. Okay, so this is when you'd get your IgG or IgA isotype of antibodies being produced. Okay, what we call the T-dependent B cell activation. So there's this CD40 ligand deficiency, or even CD40 deficiency is a type of primary immunodeficiency where patients either lack CD40 or they lack CD40 ligand. Okay? So because of this, they have immunocompetent B cells, but they can never class switch. Okay? So they tend to have very high levels of IgM. So it's the same disease that we call the hyper IgM syndrome. Okay? But almost no IgG, IgE, or IgA. So these individuals have a poor response to extracellular bacteria. And one of the things that because they don't induce class switching, they don't induce formation of long-lived memory cells, so they are prone to recurrent infections and then only ever produce IgM. Okay? The other disease etiology associated with cytokines is a defect in Fas ligand. I know you remember about the Fas ligand interacting with the Fas receptor, which is important in apoptosis of cells. Okay, either removal of autoreactive T cells, for example, during their ontogenesis. Okay, or any other way by which we want to eliminate T cells. So patients who have a defect in the gene that codes for the Fas ligand are not able to get rid of autoreactive T cells. This is very important during the ontogenesis of T lymphocytes in the thymus. You can go and revise the processes that occur there. So because of this, these patients tend to be prone to lymphoproliferative disorders, okay? So because you have all these lymphocytes accumulating in their lymphoid organs, and disorder, because they're not able to get rid of autoreactive lymphocytes, they're also prone to autoimmune disease. So they have this disease etiology. It's also a primary immunodeficiency called the autoimmune lymphoproliferative syndrome. Okay, role of cytokines in infectious disease. We've talked about that, and I've alluded to the fact that you may have disease entities where the cytokines are very important. So a good example of this is in HIV. We know the receptor for HIV entry into our cells is CD4, but there are other receptors that act as co-receptors, and these are chemokine receptors, CXCR4 and CCR5. And I'm sure you've heard about patients who have a deletion in the CCR5, which means they cannot, they don't have a proper CCR5. There's a deletion in the gene, so they don't express CCR5, meaning they lack this co-receptor, so they do not get viral entry. So these are individuals who are basically resistant to HIV infection. I think this is my last slide on cytokine assays. Basically, how can we measure this levels of these cytokines from tissue? So this can be done in biopsies, isolated cells, or in body fluids, but usually in a research setting, and even just for the measurement of blood levels of cytokines, this is not done routinely in clinical practice. Okay, and I mentioned that for the the cytokines of the adaptive immune response, they're hardly produced in large quantities that will be detectable in blood. It's only the ones that of the innate immune response in the context of a very severe infection or a case where you have massive production of these cytokines. That is when you can have measurable levels. And so that usually would be TNF or interleukin-6. Clinically, we've seen interleukin-6 being measured, but this is only in the context of SARS-CoV-2. Before we had this pandemic, really there was no one measuring interleukin-6 in blood. Okay, the different assays that can be used to measure cytokines. I think Dr. Walong, for his dissertation, for his MEd, was measuring some cytokines in genitourinary secretions. I think in vaginal fluid, he used flow cytometry. We'll go into details of these different lab methods. We can use ELISA. So for example, the interleukin-6 assay is an ELISA-based assay. The other assays like Luminex bead assays, which are basically, they combine the utility of flow cytometry and ELISA, because they use beads that have the antigen or antibody coated on that. I think only KNH has a Luminex assay, but this is for the transplant lab, which is still hasn't been commissioned. There's an ELISpot assay. We'll discuss all this, which reminds me, I need to add ELISpot onto your lecture schedule. I'll tell Dr. Oyaro to include that, because there's another ELISpot assay that is used, or a similar assay that is used to, as the in vivo method of the MAN2 test. So it's similar to the MAN2 test, but this is done in the lab as opposed to the MAN2, which is done in vivo because the injection is done on the patient. So maybe you can remind, you can ask Dr. Oyaro about the interferon gamma release assay, just to also give you the principle of that, but I'll also remind him. So there are various ways this can be done, and we'll discuss some of these methodologies, because they're not specific to cytokines, they're used for many other assays. In terms of therapy, you can either use cytokines to stimulate or enhance an immune response. So for example, interleukin-2 can be given to, so this is a monoclonal antibody, okay? Or a recombinant fashion of this cytokine to increase T-cell proliferation. So of course, you're giving it so that you enhance the effect of that cytokine in the patient. So interleukin-4 can be given to increase B-cell activation, interferon gamma to increase antigen presentation, or even to increase macrophage activation. For example, there's another group of immunodeficiencies where patients lack interferon gamma, and we'll talk about that later. Okay, the most common way we use it in KNH is when we infuse patients with GCSF, usually cancer patients who become neutropenic because of chemotherapy, like our leukemia patients, very common in our childhood leukemias. So as long as it's not an acute myeloid leukemia, so for the ALL, usually they get GCSF, Neupogen, just to ensure you can boost their neutrophil levels. The other way you can use cytokines for therapy is to suppress immune responses. So this would be cytokine blockade. We'll see the role of anti-CD25. CD25 is the CD name for the interleukin-2 receptor. Okay, remember I talked about, I mentioned that we'll talk about the T-cell activation in context of the co-stimulatory signals in transplant immunology, and we'll see where this applies, where you can actually use these molecules to block activation of your T-cells so that you can block your rejection responses. I alluded to the use of anti-TNF in rheumatoid arthritis. The other way is to just alter the cytokine balance so that you have more of the anti-inflammatory than the pro-inflammatory. So using chronic inflammatory diseases like in autoimmune disease. So this table just summarizes some of the cytokine-based therapies. The first column is the trade name, like I mentioned, Neupogen, which is actually a trade name. And the second column describes exactly what that agent is. And then the third column is the clinical application. Okay. And of course, now you know about tocilizumab, which is being used to block the action of interleukin-6 in patients with cytokine storm induced by SARS-CoV-2. Okay, do we have any questions? Sorry, I went over time.